at mitosis and the cell cycle. So mitosis and binary fission are two ways that cells divide. Mitosis usually occurs in eukaryotes, and binary fission usually happens in prokaryotes. So here's the mitosis cycle in eukaryotic organisms, and this is very familiar as far as what you probably learned in Biology 9. And this is a simple diagram of binary fission that happens in prokaryotes. Maybe not as familiar with it, but we're going to spend some time with it today. This will go pretty quickly since there's not a lot of additional material from what you probably had, like I said, when you took general biology. So first, why do cells divide? Well, cells in multicellular organisms, so like plants and animals, divide to produce more cells for an organism. So primarily they're dividing to increase cell number, and that might be needed to grow, to reproduce, to repair, or to replace. So in each of these cases, 1, 2, 3, 4, more cells are needed within a single organism. In prokaryotes that undergo binary fission, cells divide to reproduce. So when you have a bacteria and it goes through binary fission and you get 2 bacteria cells out, these are 2 separate organisms. So in prokaryotes, cell division, binary fission, is how they reproduce asexually. They make a whole new organism. So to be able to divide, we need to duplicate everything that's in the cell. So for a eukaryotic organism, all of the organelles need to be duplicated, because we can't have one cell without any ribosomes. It wouldn't function. It wouldn't work. Can't have a cell without any nucleus. It couldn't function. Wouldn't work. So that also means they need to duplicate their DNA, because one cell can't have no DNA or different DNA than the other cell. One of your skin cells can't be lacking DNA or have different DNA than all the other skin cells. That wouldn't end up very good over the long term. So the first part of mitosis, before the cells can actually divide, we need to duplicate our DNA. So both daughter cells, 2 cells that are resulting from cell division, need exact copies of the same DNA. So if I have a cell, and then mitosis happens, and we have 2 new cells when this divides, these are called daughter cells. DNA is stored in long, thin strings called chromatin, and then condenses into short bars called chromosomes when mitosis is going to occur. So normally, when your cells are not dividing, the DNA in the inside, it looks just like long strings, like that, and that's called chromatin. Only when the cell is ready to divide do they condense into chromosomes that look like this, like long sticks or bars. We as humans have 46 chromosomes, which means we have 23 pairs. 1 of each pair comes from each parent. So 1 of your pairs comes from your mom, and 1 comes from your dad. When mitosis is about to occur, chromosomes duplicate and create an X shape linked in the center. And that center, where the 2 duplicated chromosomes are linked, is called the centromere. So, for example, Here's a chromosome before it's duplicated. Once it's duplicated, it forms this X shape. In the center portion where it's linked is called the centromere. Each of the identical chromosomes that are linked by the centromere are called sister chromatids. And when mitosis happens, we separate the sister chromatids, and one goes to each new cell. So each new cell has an exact same copy. Here's a picture of all 46 of our human chromosomes. We have 2 copies of each chromosome. No, I didn't say 2 identical copies. We have 2 chromosome 1s, 2 chromosome 2s, 3s, 4s, 5s, all the way up to 2 chromosome 22s, and 2 sex chromosomes. This is a male because it has an X and a Y. When mitosis happens and they duplicate, our chromosomes will look like this. So we have duplicated both of our chromosome 1, so we have 2 X's linked at the centromere. All the way up to now we have 2 copies of each of our sex chromosomes linked at the centromere. So this is what duplicated chromosomes before mitosis would look like. The cell cycle is the pattern of cells going in and out of mitosis. So cells are actually dividing only very rarely. Only about 10% of the lifetime of a cell is spent in mitosis. The rest of the time they are just growing, doing normal cellular processes, and this is called interphase. And we can kind of split up interphase into 3 parts. The first part, G1, they're just growing and doing, I'll just write cell stuff. So normal cell processes. And then they go into S phase, or the DNA synthesis phase. This is where they duplicate DNA. And after S phase, they go into another G phase, where they prepare for mitosis. And then after G2 phase, they have their duplicated DNA. They can go to M phase, or mitotic phase, where they actually divide. Alright, the first part of the M phase, or the mitotic phase, is called prophase. In prophase, the chromatin condenses into chromosomes. So the long string of DNA, like in this picture, it's just a massive string, condenses into chromosomes. So we can see them, they look like X's, they're linked at their centromeres right here. Our centrosome duplicates, so now we have 2 centrosomes. And these long microtubule things called spindle fibers emanate from them. These spindle fibers are actually going to eventually connect to the centromere. Each spindle fibers from each of the centrosomes are going to attach to those centromeres, and they're going to end up pulling the chromosome apart to opposite sides of the cell. So that's what happens in prophase. Next is metaphase. The nuclear envelope, or the nuclear membrane fragments, becomes no more. And that is to allow the spindle fibers to attach to the chromosomes at a place called the kinetochore. Now the kinetochore is just a protein that's on either side of the centromere where the spindle fibers can attach and anchor on. So that's what the kinetochore is. The spindle fibers from each of the 2 centrosomes attach to each pair of sister chromatids at the kinetochore. So now the centrosomes are on the opposite side of the cell, and spindle fibers from both centrosomes attach to opposite sides of the same chromosome. So there's this kinetochore, and just like this one, both centrosomes are linked on. Our chromosomes are going to line up across the middle of our cell in metaphase. Metaphase middle along this imaginary long line called a metaphase plate. And you can see that one sister chromatid is facing each pole or end of the cell, attached by a spindle fiber. Right. After metaphase, the spindle fibers are going to start to pull the chromatids in opposite directions. So, each spindle fiber connected to one of the sister chromatids at the kinetochore are going to start to shorten, and the sister chromatids are going to separate, are going to start going to the opposite end of the cell. And this is anaphase. Anaphase, away, the sister chromatids are going to move away from each other. As the chromosomes reach the opposite side of the cell, two new nuclear membranes are going to begin to form around those chromosomes. So nuclear membrane will start to form around these, and nuclear membrane will start to form around these. That is telophase. So telophase, you've got one centrosome, one nuclear membrane with one set of chromatins starting to form on each cell, and the plasma membrane that was around all of this starts to pinch off in the middle. This is called a cleavage furrow, and what's going to happen is it's going to pinch off and form two new cells. So the act of it pinching off and separating is called cytokinesis, or the separation. And that's how we get two new cells. It'll pinch off right here, two brand new cells. Right. So that was basically all the important steps of mitosis for multicellular organisms. In binary fission in bacteria and prokaryotes, any asexual reproduction, binary fission happens. It occurs pretty much the same as mitosis did, except there's some differences. One is that there's circular DNA in bacteria, and we need to replicate that too, and it starts replicating at an origin of replication. So say we have this bacterial DNA, this plasmid, we're going to replicate it. What happens is a small bubble forms in that plasmid. This is going to be our origin of replication. And the DNA is going to start replicating in both directions and separating it into, let's see if I can draw this. Two circular plasmids within our cell. And as it replicates in both directions, two plasmids will be formed. And the plasmids start migrating to the opposite side of the cell, just kind of like an anaphase in mitosis. And the cell membrane starts to pinch off, like in telophase. But you remember that in bacterial cell walls, bacteria, there are cell walls. So a new cell wall is going to start to form right in between here, what we call a cell plate. And that is actually going to materialize and cut the cells into two, the two bacterial cells. Basically, all that binary fission is. We're making two new plasmids. They go to the opposite side of the cell, and membrane pinches off, and a new cell wall is made, new cell plate is made, separating them, the bacteria. Alright, that is pretty much it for, that is it for mitosis and binary fission. Not very much new, but I wanted to go over it so you can kind of see the timeline from interphase, prophase, metaphase, anaphase, telophase, and binary fission. So 9.1, 9.2.